All right, welcome to the Explainer. Today we're diving headfirst into some massive AI news that is absolutely shaking up computational biology. We're looking at a brand new paper accepted at the 2026 ICML workshop on generative and agentic AI for biology that introduces something called Design CP. This is a total game changer because it fundamentally shatters the single GPU memory limits for generating large protein structures using RFdiffusion 3. If you've ever tried designing massive multimeric protein assemblies and just sat there watching your hardware crash, well, you know the pain, and this explainer is exactly what you need.
Okay, let's dive into this. Here's our roadmap for today. We'll start with the protein design bottleneck, introduce Design CP, look at how context parallelism actually works, compare 1D and 2D sharding strategies, cover the hardware and deployment needs, and finish up with how this is democratizing large protein design.
So, Section 1, the protein design bottleneck. Let's look at why your hardware keeps crashing.
It really is the ultimate frustration for researchers in this space, right? You have these incredible all-atom generative models like RFdiffusion 3, which theoretically can design large multimeric assemblies by jointly modeling all the chains together. But in practice, you try to run it and bam, you get hit with an out-of-memory error. The GPU simply taps out. So why exactly does this happen? Well, the main culprit is something called quadratic activation memory. You see, the real problem isn't actually storing the model's weights. The real bottleneck is the dense, pairwise representations that these diffusion models absolutely need to capture structural dependencies. Literally every forward pass computes attention over all token pairs, and those resulting activation maps, they have to live in your GPU's VRAM. As your sequence length and chain count grow, the math gets brutal, fast.
What's really interesting about this slide is the harsh reality of that quadratic scaling. If you simply double the number of residues in your design, your activation memory footprint doesn't just double, it quadruples. It expands by 4x. This right here is exactly why single GPU limits cap our feasible designs at very modest subunit counts. You basically have two choices. Either your system crashes, or you're forced to use aggressive gradient checkpointing, which, let's be real, absolutely tanks your throughput.
Section 2. Introducing Design CP. We're slicing the problem rather than the model. Enter Design CP. This method elegantly shifts the bottleneck completely away from weight storage and over to activation distribution. The absolute brilliance here is that we are distributing those massive quadratic memory activations across a multi-GPU mesh without touching the model's brain. You don't need to modify or replicate the pre-trained weights at all, you're just sharding the context, meaning the data being processed rather than the model itself.
So what does this mean for you? Well, it gives you three massive benefits. First, you get to distribute those heavy quadratic activations across multiple GPUs that are all working together. Second, there's absolutely zero retraining, no weight adaptation, and no custom diffusion schedulers required. And third, and this is huge, it keeps the diffusion sampling loop mathematically identical to the single GPU baseline. You are getting the exact same pre-trained diffusion trajectory, it's just spread across more hardware.
Let's move to Section 3 and see how this builds. How context parallelism works. Let's look at the mechanics. Now, context parallelism is fundamentally different from traditional model parallelism. Imagine you're trying to build a massive, highly complex puzzle. Instead of trying to cram the whole puzzle onto one tiny table, or giving everyone their own duplicate puzzle to work on separately in different rooms, you just push several tables together. You are partitioning the sequence dimension itself across the devices.
And here is the step-by-step choreography for how that actually works. Step 1. Each GPU in your mesh takes a specific subset of the sequence tokens. Step 2. Each GPU then computes the local attention for its specific subset. And step 3, to make sure the entire protein complex actually makes sense as a whole, a mechanism called ring attention coordinates the cross-partition communication. It synchronizes everything seamlessly, which preserves the pre-trained weight layout without introducing massive sync overhead.
Section 4. 1D versus 2D sharding. Let's compare the specific strategies Design CP brings to the table.
So the researchers actually offer two different flavors here. On the left, we have 1D row sharding. This is kind of your flexible option. It works great if you have linear GPU chains or slightly mixed heterogeneous clusters. But, it does trade a bit of efficiency for that flexibility. On the right, we have 2D grid sharding. This uses ring attention organized in a logical ring topology, which moves gradient and activation sinks in a single pass around the mesh. It aligns beautifully with the rectangular communication patterns of diffusion models, which gives you much lower cross-GPU latency. The catch? You absolutely need a strict rectangular GPU layout.
When the researchers benchmarked these strategies on icosahedral assemblies, they looked closely at the maximum feasible asymmetric subunit size. The crucial point here is that square root scaling trend. Because the memory scales quadratically, when you distribute it linearly across n number of GPUs, the max sequence length you can actually support only scales with the square root of n. To put that into perspective, practically speaking, this means if you want to double your target residue size, you're going to need roughly 4 GPUs.
Section 5. Hardware and deployment needs. Time for a reality check on what a developer actually needs to know to deploy this today.
Honestly, the deployment is remarkably clean because DesignCP operates strictly at inference. Step 1, you literally just configure the backend to match your GPU mesh, whether that's 1D or 2D. Step 2, you apply your point group symmetry constraints, and this brilliantly illustrates one of the best features of this whole paper. These constraints are baked right into the forward pass before the partitioning even happens. The mesh handles symmetry expanded activations automatically, completely eliminating those tedious iterative refinement loops. And step 3, you just run the standard RFdiffusion 3 pipeline. No custom Docker images, no weird proprietary runtimes, just standard PyTorch distributed inference.
But, as with any deployment, you really need to watch out for a few edge cases. First, if you run asymmetric designs without symmetry constraints, you are still going to hit that square root scaling limit. Remember, context parallelism distributes the quadratic law. It doesn't just magically erase it. Second, if you use ring attention, you absolutely must ensure you have contiguous GPU IDs or an explicit topology mapping. If your ranks are misaligned, you will get silent synchronization failures, and let me tell you, those are an absolute nightmare to debug. Finally, keep an eye on your memory ceiling.
And on that exact note, you should always leave about 10 to 15% of your VRAM as headroom. If you push the asymmetric subunit size right up to the absolute memory ceiling of your cards, the in silico structural and interface metrics actually start to degrade. So give the activation checkpointing the breathing room it needs to function properly.
Finally, Section 6, democratizing large protein design. Let's step back and look at why this is such a big deal for the field.
16 gigabytes. Honestly, this is the detail that truly blew me away. The authors didn't just prove this method on some massive multi-million-dollar supercomputer. No, they successfully demonstrated end-to-end all-atom design of octahedral nanoparticles on a small cluster of standard workstation-grade 16 gigabyte GPUs. This completely changes the game. You no longer need exclusive access to an enterprise data center to do cutting-edge multimeric protein design. The workflow yields highly favorable in silico structural metrics without needing any post-hoc alignment or manual constraint tuning. It is just a robust end-to-end process for incredibly complex structures, like icosahedral nanoparticles, running on hardware that a mid-sized lab or even an ambitious startup can easily string together.
As the researchers so perfectly put it in their abstract, DesignCP can be a practical path towards democratizing large assembly protein design. By distributing the computational weight, they are essentially unlocking the full generative potential of RFdiffusion 3 for a vastly wider community of scientists and engineers.
So we've seen how context parallelism neatly bypasses the single GPU memory bottleneck, keeping the math exactly identical while unlocking unprecedented scale. The barrier to entry for large-scale all-atom modeling just drops dramatically. So the only question left to ask is this: what breakthrough complex are you going to design now that the hardware limit is finally gone? Thanks for joining me for this explainer, and keep exploring.